I need to.
Good evening. My name is Don Johnston, and on behalf of the Ray Dorough Speaker Series Board of Directors, I welcome you to this evening's live stream featuring neurologist Dr. Richard Reimer. Tonight's live stream is a departure from our usual format in a couple of ways. We are privileged to partner with the Livermore Lab Foundation to bring you this important discussion of neurodegenerative diseases. Amyotrophic lateral sclerosis, ALS, also called Lou Gehrig's disease, Alzheimer's disease, and Parkinson's disease. Proceeds from this evening's live stream will benefit the Foundation's ALS research initiatives. We also would like to note that April is, very appropriately, National Parkinson's Awareness Month. Here's how we will proceed this evening. I will introduce Donna Crawford, President and Chair of the Livermore Lab Foundation. After a few words about the Foundation's work, Donna will introduce former Livermore Mayor Kathy Brown, who will give us a personal testimonial on her husband's battle with ALS. Donna will then introduce Dr. Reimer. Please remember that at any time during the presentation, you may submit questions via chat. At the conclusion of Dr. Reimer's talk, he will answer as many of your questions as we can get to in the time allowed. I'd now like to introduce Donna Crawford. Donna Crawford is the founding chair of the Livermore Lab Foundation, incorporated in 2016. Prior to retirement and heading the foundation, Donna Crawford served as the Associate Director for the Computation Directorate at Lawrence Livermore National Laboratory for 15 years. During that time, she led the effort to develop high-performance computing capabilities at the laboratory, one of the world's premier scientific computing centers. As Associate Director, Donna was a strong advocate for science education and training, particularly for underrepresented populations in the scientific community. She also sought opportunities to apply the laboratory's unique capabilities to research benefiting society. As chair and president of the Livermore Lab Foundation, Donna continues this very important work. Please welcome Donna Crawford.
Thank you, Don. Good evening. The Livermore Lab Foundation is delighted to sponsor tonight's presentation. We're dedicated to advancing science and inspiring next generation scientists. And we do that by leveraging the incredible expertise and capabilities at Lawrence Livermore National Laboratory. The lab, as many of you know, tackles important, complicated, complex problems that demand the best science. And that science is often applicable to societal problems. That's where a nonprofit like Livermore Lab Foundation comes in. We provide opportunities for private foundations, philanthropists, and donors like you to advance that important work. Your purchase of tonight's ticket helps contribute to the lab's ALS research. So let me take a moment to say thank you to all of you for joining us tonight. When our foundation launched in 2016, healthcare was identified as an important focus area. The lab works on healthcare in as much as it relates to national security, using its high-end data analytics, supercomputers, and computational modeling, along with bioassays, bioengineering expertise, and high-resolution brain mapping. The foundation leverages those capabilities for ALS research. Bringing the lab into the fight against ALS has the potential to improve the world's understanding of this deadly disease and consequently accelerate the development of new therapies and treatments. The lab is interested in taking on this project because it helps build out their broader neurodegenerative disease capabilities. While working on one of our projects, we had the privilege of meeting tonight's guest speaker, Dr. Richard Reimer. However, before he begins, we've asked a familiar face, Kathy Brown, to bring this down to a personal level for us. And I know there are many of our viewers here tonight who've also been touched by devastating neurodegenerative diseases. Let me introduce Kathy. Kathy Brown is the former mayor of Livermore and longtime supporter of the lab and the community. She was a founder and director of the Tri-Valley Haven for Women in Livermore, City of Livermore Planning Commissioner, founder of Livermore Youth for Action, and executive director of Alameda County Project Intercept, just to name a few. She was also the first American mayor to visit a former secret city in Russia. No small feat for someone raised in the shadow of the Cold War. Mayor Brown holds a bachelor's degree in criminology from UC Berkeley and a Master of Public Administration from Cal State Hayward. And regrettably, she lost her husband, Tom, of 53 years, to ALS last August. Kathy?
Thank you for that kind introduction, Donna. I'm honored you've asked me to join you for tonight's event, and equally honored to be here to both listen and learn from Dr. Richard Reimer, a renowned neurologist from Stanford. ALS is a really tough subject. It's tough to talk about, it's tough to understand the randomness of who it afflicts, and it's tough to watch your loved one go through all the stages of this horrendous disease. As Donna mentioned, I know this from experience. In fact, let me introduce you to this guy. My husband of 53 years, Tom Brown, died in August from ALS. It was a brutal journey. Oops. This cutout of Tom was created by Mike Piscotty's ALS team at the Oakland A's, where his son plays. I'm thinking right field, but hopefully I have that right. Tom was in the stadium during the last baseball season as a cutout. When Tom was diagnosed, we were told there was no treatment and it's fatal. He is one of 20,000 individuals who are part of the statistic of this formidable enemy. And I'm sure some of you joining this event tonight know that journey. And that's why our commitment, our collective commitment to find a cure for ALS is so important. My family, my children, my grandchildren, and I made this commitment during Tom's own struggle with ALS. We promised him we would help other families fighting the fight, and we would take up the cause to bring additional attention to this rare disease. Put him down. During my own career, I've been fortunate to have personally witnessed the unparalleled science and research excellence that occurs at Lawrence Livermore National Laboratory. I know they are capable of taking on this grand ALS challenge to identify biomarkers, understand the influence of genetics, and help find new treatments for a cure. I'm grateful there are philanthropic leaders like Mike Piscotty of ALS Cure Project and Donna Crawford of the Livermore Lab Foundation, who are rallying others and fighting to bring more attention to our cause. And those of you who know me probably attest that I can be a bit of a fighter myself. I'm proud of that moniker because I know that's what's needed. 2020 has been a really tough year for all of us. But the ticket you purchased to attend tonight's event, as well as your presence and your interest in this subject, can and will help us on the journey to find a cure for ALS. And for that, I would simply say thank you. Thank you on behalf of Tom, on behalf of all who have left us much too early, and thank you on behalf of those who have yet to become afflicted. ALS, like I said, is a tough subject to discuss, but I dream of the day when we can hear that ALS is no longer a challenge. It will be a day when the top stories we hear in the news reflect the significant progress that has been made and the ensuing lives that might be saved. Now, I am a Cal girl at heart with the Go Bears mantra firmly imprinted on my brain. After all, Tom and I met at Berkeley. He played football for Cal and I was a pom-pom girl. But tonight, since we're well past the big game, let me give a collegiate salute to Dr. Reimer and that other university across the bay for all his team is doing on this issue. Your work is impressive, and I'm very grateful. Keep up the fight, and thank you all.
Thank you, Kathy. We're inspired by what you and your family are doing in the fight for a cure.
Thank you.
Now, let me introduce our speaker, Dr. Richard Reimer. He will begin his talk by sharing a broad overview of common neurodegenerative diseases, including Alzheimer's, Parkinson's, and ALS. Using ALS as an example, he will then present the evidence for the roles of aging, genetics, and the environment in the disease onset and progression. Dr. Reimer is an associate professor in the Department of Neurology and Neurological Sciences at Stanford University School of Medicine, and a staff neurologist for the Palo Alto Veterans Affairs Healthcare System. After obtaining his undergraduate degree at Yale University and receiving his MD from Emory University, he completed his neurology training at UCSF. His primary research interest is in defining the mechanisms underlying neurological diseases, with the goal of identifying novel sites for therapeutic interventions. The research Dr. Reimer conducts ranges from the study of mouse models for rare lysosomal storage disorders to the machine learning-based analysis of electronic health records of ALS patients to identify risk factors and disease modifiers. Without further ado, please welcome Dr. Richard Reimer.
Donna, thank you very much. And Kathy, thank you for putting my talk in context that appeals that everyone can understand. And Don, thank you also for inviting me to speak today. So I'm going to start by sharing my screen, and we'll get started here. All right. So as Donna mentioned, I'm going to talk today about ALS, Alzheimer's disease, and Parkinson's disease. And the title of my talk is Who's at Risk? I'm going to talk about aging, genes, and environment, and how they contribute to these diseases. This is an overview of my talk. I'll begin with a brief description of these diseases, and then provide evidence for aging, genetics, and environment in each of them, but briefly. And then I'm going to go do a deeper dive on ALS and look at the risk factors and how they contribute to the disease. And then I'm going to talk about a machine learning project that I'm involved in with members of the Lawrence Livermore National Lab. And then lastly, I'm going to finish up with an optimist view. I'm the optimist view of the future for ALS and other neurodegenerative diseases. So let me start with a brief description of these diseases, and I will mention how each of them ties together as a group. So these diseases are all common, and we all probably know someone with one of these diseases. Alzheimer's disease is the most common, with 5 million people in the United States affected. Parkinson's disease, 500,000. And ALS is rare. There are about 20,000 people with the ALS at this time in the United States. These are all diseases that affect the brain and are more common with aging. And these are the diseases that we treat as neurologists. And I'd argue that the dilemma of the neurologist is that there are no truly effective treatments that significantly affect the course of these diseases. And this is what I really hope to change in the work that I do. So we'll start with Alzheimer's disease, and I'm going to give you a brief clinical description. Dementia is a progressive decline of cognition that affects memory, judgment, language, visuospatial function, and personality. Dementia has several causes, but Alzheimer's disease is the most common cause. And here I have some pictures of brains from autopsy. On the left is the brain of somebody who died in their 60s, who did not have dementia or Alzheimer's disease. And you can see that the brain is full, it's tightly compacted. And over on the right is a picture of the brain, of a section of the brain from somebody who had Alzheimer's disease. You can see that the space in between the different parts of the brain is much bigger. The brain itself is smaller, and this is typical of what we see in this disease. And this is what we see as we describe it grossly, or sort of without a microscope. But if we use a microscope, we see some classic findings in people who are affected by Alzheimer's disease. We see these brown accumulations of protein the protein is called amyloid beta, and these are these classic plaques of Alzheimer's disease. In addition, we see accumulation of another protein called tau within nerve cells that are affected by Alzheimer's disease. These are called neurofibrillary tangles. So next, the disease I wanted to talk about next is Parkinson's disease. This is also a neurodegenerative disease. It's a movement disorder and characterized by slowing of movements, tremor, stiffness, and poor balance. And on the right here, you see a drawing that was done by Sir William Gowers in 1886. And if you show this drawing to anybody who's gone through medical school, they immediately can identify this person as someone who has Parkinson's disease by the classic sort of posture of the person and the way they apparently are moving from the drawing. The disease was initially called the shaking palsy by Parkinson, and that's because there's a palsy or slowness and shaking at the same time. If we look at the brain of somebody who's affected by Parkinson's disease, and this is part of the brain called the brain stem. This is just above the base of the brain, base of the skull. And you can see this dark line here in the person who did not have Parkinson's disease. This dark line is made up of nerves in the brainstem that produce a substance called dopamine. And as these cells die, this dark pigment starts to go away. And you can see here on the right, the brainstem of a person who had Parkinson's disease, and that dark band is almost completely gone. And that's typical of what we see. And that dark substance led to the name substantia nigra for this region of the midbrain. And just like we saw accumulation of amyloid beta in the Alzheimer's disease brain, we see accumulation of another protein in these structures here, these pink dots that we see in the brain of people with Parkinson's disease. These are called Lewy bodies, and they're filled with lots of proteins, including something called alpha-synuclein. So you can think of these as aggregates or gobs of protein that have sort of gummed up the works of the nerve cell. And we see the beta amyloid in the Alzheimer's disease, the neurofibrillary tangles in Alzheimer's disease, and then these Lewy bodies in Parkinson's disease. Interestingly, we think of Parkinson's disease as a movement disorder, but it also affects cognition. And 30 to 40% of patients with Parkinson's disease have dementia. Lewy body dementia is associated with the same pathological findings, those Lewy bodies that I showed in the previous slide, as we see in Parkinson's disease, but the Lewy bodies are present not just in the substantia nigra, but throughout the brain. Next, I wanted to talk about, give you an overview of ALS. So ALS, or amyotrophic lateral sclerosis, is also called Lou Gehrig's disease. It was named that because Lou Gehrig, a baseball player for the Yankees in the 1920s and 1930s, was affected by the disease and died at a young age. But he's not the only person who has ALS. Many people do, including Stephen Hawking. So what is ALS? ALS is another neurodegenerative disease, and it affects motor neurons. These are the nerves in the brain and the spinal cord that contribute to or allow us to move. The neurons are in the spinal cord, the motor cortex, and also in the brainstem. The loss of these motor neurons causes a relentlessly progressive weakness and a loss of muscle mass. So here in these photographs, you can see the hand of a patient who's affected by ALS. On the top picture, you see that the base of the palm is kind of flat. And if you look at your own palm, you might see that there's a big bump there. That's a big giant muscle. And as one develops ALS and starts losing muscle mass, these are some of the muscles that you see disappear. And in the lower picture, you see that the space between the thumb and the index finger is kind of hollowed out. And that's, again, caused by the loss of the muscle mass here. So that's where the amyotrophic means loss of muscle, comes from in the name. And then the name lateral sclerosis comes from what we see in the spinal cord. So this is a section of spinal cord from somebody who had ALS. The motor neurons that are in our brain that allow us to move, say if I want to move my right arm, there's a motor neuron in my left motor cortex that sends a signal down to the spinal cord, and it sends a signal through the lateral corticospinal tracts, these regions right here that are outlined. Those contain these fibers that come from the neuron in the brain. They then connect with motor neurons in the spinal cord, the second set of motor neurons that we call lower motor neurons. And these lower motor neurons then go out to the nerve. And you can see that depicted here. This is a picture of a person's spinal cord at autopsy. And you can see that this is an interesting picture of somebody who had ALS. You can see these lower spinal nerves, these are the nerves that are coming out of the spinal cord, are very thin and kind of wispy. And the upper ones seem much thicker, and you go up a little bit higher, you see even thicker nerves there. And what has happened here is this person has ALS that's affecting the lower portion of their spine, but not so much in the upper portion. And that's pretty common in ALS, that we'll see people who initially the disease may start in their legs and then slowly starts to creep up. And we think that it's actually progressing within their spinal cord. So this here, I wanted to emphasize that you see this lateral sclerosis, this thinning and scarring of the lateral corticospinal tracts, and that's where the lateral sclerosis portion of amyotrophic lateral sclerosis comes from. And just like we saw in Alzheimer's disease, the plaques and tangles that we saw there, and the Lewy bodies that we see in Parkinson's disease, we see aggregates of proteins in ALS. And those aggregates are mostly composed of a protein called TDP43. And that's immunostained here in this person who has had ALS. This is a section of a spinal cord from somebody with the ALS. And just like we saw in Parkinson's disease, which is a movement disorder, in ALS that we think of as a disease that really affects motor function more than anything else, we also see dementia. So about 40% of patients who have ALS have some cognitive impairment, and more than 10% have a diagnosis of something called frontotemporal dementia. And frontotemporal dementia is a particular type of dementia where behavior seems to be a predominant factor rather than, say, memory, as we see in Alzheimer's disease. Within families that have ALS, and ALS, I'll talk about that a little bit more, one of the genes that can cause this is a gene called C9orf72. And this gene variant can affect members in the same family different ways. So some family members may have ALS, some may have FTD, and some may have both. And some may not have anything at all. And interestingly, the TDP43 pathology that I mentioned, in other words, that accumulation of TDP43 that we see in ALS patients, we see in people who just have FTD, whether or not they have a family member with ALS. So these diseases, ALS, Alzheimer's disease, and Parkinson's disease, are clinically and pathologically distinct, but they're all hallmarked by the accumulation of aggregates of proteins and neuronal cell loss, and they can all affect cognition. So I think that we should really think about lumping these diseases together rather than splitting. And this lumping and splitting is a common theme in medicine. Some people like to lump diseases together, and some people like to split them into individual diseases. And I think for the sake of trying to understand these diseases better, it's probably good to think about how we can lump them together and look for commonalities and understand these diseases as a group rather than each individual disease. And if you look at pathology in people who have these diseases, not surprisingly, we see a lot of overlap. So in these two pie charts that we have here, we're going to be comparing the distribution of these different accumulation of these different proteins, the beta amyloid and tau that we typically see in Alzheimer's disease, the Lewy body alpha-synuclein that we see in Parkinson's disease and Lewy body, and then the TDP43 that we see in people with FTD and ALS. If you look at somebody who has Alzheimer's disease, you'll often find alpha-synuclein, TDP43, or TDP43 and alpha-synuclein accumulations. In fact, pure tau and amyloid beta accumulation is rare in a patient with Alzheimer's disease. And if you look at somebody who has Lewy body disease or Lewy body dementia, again, it's rare just to see the pure alpha-synuclein accumulations. You'll often see TDP43 or amyloid beta. So it suggests that these diseases are common in terms of the risks that they may have, but also common maybe in how the diseases progress. So I want to now briefly discuss the evidence for aging, genetics, environment in these three diseases. I'm just going to show you a chart, but or rather a table, but I want to use it as an overview, but we'll go into more detail on ALS later. So when I think about risk factors, I like to think about them in groups, the nature group and the nurture group. The nature group is the genetics. So what genetic variants have we inherited from our parents, and how might these be associated with disease? Another aspect of nature is aging. So we all age. The clock is ticking on all of us. And it's important to think about whether or not a particular disease has some association with age. So obviously there are diseases that affect children, but there are also diseases that affect adults. And there's some diseases that get more common as we get older. And these three diseases fall into that category. As for nurture, I think of this as the environment. What do we get exposed to? What's impacting us as we get older, or as we live our lives? So one thing I think it's important is that when we study risk factors, we're really looking or identifying associations, not causation. And if you think about this, and think about all the reports on the different diets and which diet is best for your health, you can understand how interpreting association as causation can be a problem. So it seems like each year there's a new diet that works better than the last year. And that's probably because there was an association with health, but not necessarily an understanding of the causation of how a particular diet improved health. So I would argue that when we look at this table that I put together here, we should take it with a grain of salt. But I think it is informative if we look at these diseases as a group and ask a couple of questions. So the first is, do these diseases increase as we get older? And the answer is yes for all three of them. The next question is, are there hereditary forms? In other words, are there forms of the disease that seem to run in families? And that's true for all three of these diseases. It's not the most common form of the disease, but it is true for each of these diseases. And each disease has at least one very dominant gene that's been associated with the disease. For example, in Alzheimer's disease, it's a gene called ApoE. So most of you may know or may have heard about ApoE4 as a risk factor for Alzheimer's disease. That's the most common risk factor that we know of genetically. And then for Parkinson's disease, there are several genes that have been identified that confer risk for the disease as well. And GBA is probably the most common. For ALS, I think I already mentioned the C9orf72 gene. That's the most common gene that we know is associated with ALS. But I think perhaps the more intriguing question is, are there environmental risks that seem to increase the likelihood that someone might develop one of these diseases? For Alzheimer's disease, there's been a lot of study on this, and there have been several different factors that have been identified that increase or decrease your risk for getting the disease. For increased risk, we know that diabetes, high blood pressure, high cholesterol, all contribute. And these are the risk factors for heart disease as well. And an interesting thing is that there was a lot of concern that the number of people with dementia is going to increase astronomically as our population gets older. That hasn't really turned out to be as worrisome as we thought, in part because we've been treating heart disease much more aggressively. And in doing so, we've actually been reducing the risk for people getting dementia. Hearing loss is another risk factor for Alzheimer's disease, as is TBI, traumatic brain injury, pollution, and smoking. And you can probably combine pollution and smoking together. In terms of risk factors or things that decrease your risk for developing Alzheimer's disease, we know that education, so higher level of education, seems to protect you from developing Alzheimer's disease. Social contacts, the more social contact that you have, the less likely you are to develop an Alzheimer's disease. And interestingly, physical activity seems to reduce your risk for Alzheimer's disease as well, particularly physical activity later in life. Parkinson's disease has its own set of environmental risks. Pesticides and dairy, which are associated with rural upbringing. So where you live when you're growing up seems to increase, may affect your risk for developing Parkinson's disease. TBI, or traumatic brain injury, again shows up for Parkinson's disease. And interestingly, melanoma, the cancer melanoma, is associated with an increased risk for Parkinson's disease as well. The factors that decrease your risk for Parkinson's disease are really kind of interesting. Smoking and caffeine consumption are surprisingly factors that decrease your risk for Parkinson's disease. There's a lot of ongoing research on this, and there are a number of theories. Most people don't think that smoking is actually to decrease your risk for Parkinson's disease, but they think that people who don't smoke are more likely to get Parkinson's disease because of a behavioral component. So it may be that someone who's going to go on to develop Parkinson's disease is less likely to develop habits that are addictive. So smoking and caffeine are two of the more common forms of addiction that we see in people. And then again, physical activity, just like it does for Alzheimer's disease, decreases your risk for Parkinson's disease. For ALS, we know less, and this has to do with the fact that the disease is much less common. But we do know that smoking increases your risk for ALS, as does low body mass. And this is an interesting thing, the low body mass. That may be that people, as they develop ALS, they start to lose body mass. And so by the time that they're diagnosed, they've actually lost some weight due to the disease itself. But physical activity has been listed as a risk factor for ALS in several different publications and several different studies. And I will go into that in more detail later on. Unfortunately, we haven't identified any factors that seem to reduce your risk for ALS. So now what I'd like to do is review the risk factors that we do know about for ALS, or which studies have uncovered as potential factors that have influence how likely you are to get the disease. So we'll first look at the role of aging in ALS. And this is a graph from Morbidity and Mortality Weekly Report. So this is a publication from the CDC. And in 2018, they looked at, over the previous 4 years, the distribution of patients with ALS by their age. So you can see people who are young, 18 to 39-year-olds, have a very low number. There is a small number of patients that have that disease. When you get to the 40 to 49-year-olds, it goes up and continues to go up till you get to the 70 to 79-year-old group. And this is per 100,000 people in that given population. So this is sort of age adjusted, if you'd like. Interestingly, once you get to the next group, those over 80, the number of patients goes down. And there are two potential explanations for this, and they're both probably at play. First is that if you're over 80 and you develop ALS, your life expectancy is a little bit shorter. So you're going to end up on this graph for a shorter period of time. The other possibility is that if you're at risk for ALS, it's going to declare itself at a younger age. So if you've gotten to the age 80 and you haven't gotten ALS, you probably don't have some predisposition, or you probably haven't been exposed to some factor when you're younger that is going to increase your risk for the disease. And if you look at the graphs, similar graphs for Parkinson's disease, you see the same exact pattern of this increase until you get to 70 to 79, and then it starts to drop off. For Alzheimer's disease and dementia, it's not quite the same. So it seems that as we get older, we continue to have increased risk for the disease. And that just may reflect differences in the way that the disease progresses, or possibly risk factors for the disease. So next, I want to talk a little bit more about the genetics of ALS and the genes that seem to increase our risk for the disease. So about 5% of patients have a clear family history of ALS. And when it runs in families, it runs in what we call an autosomal dominant fashion. In other words, one copy of the gene, we have 2 copies of all of our genes. One copy of the gene one copy of the gene can cause the disease. So we'll see it in each generation. And there are several genes that have been identified in ALS, but the 2 I think that are the most important to remember are SOD1. That was the first one that was discovered, and that was in 1993. And the second one is C9orf72, which I mentioned a couple times already. And interestingly, C9orf72 is the most common gene associated with ALS, but it wasn't discovered until nearly 20 years after SOD1. And the reason for that is probably twofold. So one, I mentioned already that C9orf72 can cause FTD or ALS or both. So a lot of times when there is C9orf72 was in a family, it wasn't discovered as a gene for ALS because not every family member that had that gene got ALS. And the other possibility, other explanation for why it wasn't discovered until much later, is that the gene, the mutation that we see, or the variant that we see in this gene, is an expansion of a particular part of the gene. And when you expand something, it's very hard to pick that up with the techniques and methods that we use to discover genes. So those 2 reasons probably contribute to the fact that it was discovered much later, even though it's a much more common gene. So if we next look at a pie chart for the genes that have been isolated or identified in ALS, and this is for patients who have a family history of ALS, you can see that the most common one is the C9orf72 here in orange, and then SOD1 is the next most common, and then a protein called TARDBP, and then FUS are the other genes that have been identified and are relatively common. There's some that are where we have very few patients, and they don't even make up enough to show on this pie chart. But you can also see that there's a big chunk here, about a third of the patients, where even though there's a strong family history, we haven't identified the gene yet. And that's going to continue to be true for a while, but each year there are 1 or 2 new variants that have been identified. So that's it suggests that over time, this blue section here is going to get smaller and smaller. So this is, again, the patients with a family history. Interestingly, if we look at patients with sporadic ALS and look for gene variants in these patients, we actually do find some. And in fact, we find C9orf72 about 5 to 10% of the time. We also do find SOD1 mutations, and then some of the other genes as well. But again, this is the vast majority of people with ALS are in this sporadic group. About 5% are in the familial group. And in this group that have sporadic or non-familial ALS, about 5 or 10% of the time, we can find a gene that's associated with the disease. We haven't, unfortunately, been able to find a common theme for all of these genes. Each gene seems to affect a slightly different part of the cell, but there does seem to be a theme amongst most of them that has something to do with processing of a part of the cell called RNA. So I've talked about aging and genetics in ALS. And lastly, I want to talk about the role of environment in ALS. And this is a controversial subject, and it's controversial for a couple of reasons. I think one of the main reasons is that we don't have a lot of great data on this. And that's again because ALS is relatively uncommon. And if you think about trying to look for environmental risk factors, what are the kind of complications that we run into when we do that? There's this term expososome. So this is, people talk about proteomes and genomes. Well, the expososome is what you've been exposed to over your lifetime. And this can be, and this is going to be huge in terms of space and time. So they're going to be things that you're exposed to in utero. So while you're a developing fetus, you're going to be exposed to things that they may actually have an impact on whether or not you develop a disease 50, 60, 70 years later. And then as you go through your life, think about all the different things you're exposed to, from your diet to things that you inhale to medications that you take. So it can be really complicated, and it makes the research very difficult, and oftentimes it makes it difficult to come to meaningful conclusions. The other thing to keep in mind is that environmental research often relies on time-consuming and invariably incomplete surveys. So if I were to ask you, what do you eat? You can tell me what you ate yesterday, but you can't tell me all the things that you ate over the last month. And it could be that one or two of the things that you eat infrequently may actually be the thing that's affecting your risk factor for a particular disease. And it's also important to remember that looking for environmental risk factors works well for common exposures that have large increases in risk for common diseases. And the classic thing to think about is smoking and lung cancer. And even then, it was difficult for doctors to uncover that link. And wasn't uncovered until the 20th century. So what are the things that have been linked to ALS? There's a pretty long list, but I think that these are the ones that seem to come out more often than not in repeated studies. One of them I mentioned already is smoking. Another is trauma. So physical trauma, whether it be car accidents or other things. Rural residence has also been linked to ALS. Why, we don't know. And alcohol use has been linked as well, but again, we don't know why. Military service has been linked to ALS, and that actually has to do in part with my connection to this, as I work at the Veterans Hospital here in Palo Alto. But another risk factor that's been associated with ALS in multiple studies is physical activity. And this is really a kind of interesting and controversial subject. So one of the questions that always comes up, and particularly because of Lou Gehrig's association with the disease, is there a link between sports and ALS? So here's a picture of two Yankees from Murderers, I think it was called Murderers Row. Lou Gehrig on the left and Babe Ruth on the right. They were teammates on the Yankees in the 1920s and 1930s. And in 1937, by the 1937, Babe Ruth had retired, but he continued to be in the public eye, and he made this comment about Lou Gehrig. Lou Gehrig actually was known for being the Iron Man of baseball, and he was called that because he played in over 2,000 games in a row without taking a day off. And Babe Ruth said, "I think Lou's making one of the world's worst mistakes a ball player can make by trying to keep up that Iron Man stuff. He's already cut 3 years off of his baseball life with it. The next 2 years will tell Gehrig's fate." And Babe Ruth said this in January of 1937. So was he right? So this is a graph of batting average trends for Lou Gehrig. And you can see these two years here are 1936 and 1937. And this is pretty typical of the way he would play and the way he would hit. He'd start off in the middle of sort of his average batting average, start to increase over the course of the summer as he got better and better. And knew how pitchers were to pitch him. And then near the end of the year, as most ball players do, he kind of trailed off a little bit. It wasn't hitting quite as well. 36 and 37, this is all great. And this is a deviation from his batting average between 1932 and 1935. So anything above 0 means he's doing better than he was the years before. But in 1938, so 2 years after Babe Ruth, a year to 2 years after Babe Ruth made the comment, this is what happened. So he started off a little bit lower than he had in the previous 2 years, and then started to really decline significantly. This was the last full year he played. In 1939, he started playing, but then retired in June of that year and died 2 years later. So maybe Babe Ruth knew something, and that has continued to sort of be in the back of most people's minds. Is there a link between what Lou Gehrig did for a living and the fact that he got ALS? So one way we can think about this, or we can look into this, is to use some crowdsourced information. So Wikipedia is a great source of information for most people on most topics. And they actually have a page of a list of people, of notable people with amyotrophic lateral sclerosis. There are 111 people on this list. And if you look at the occupations of these people, or in other words, why these people are notable, 40 of them are in arts and entertainment. This includes Charles Mingus and Sam Shepard. 15 are professional football players, including Dwight Clark, who many of us know of because of his connection to the Bay Area, Tim Green. There are 5 professional rugby players, 15 professional soccer players. There are 5 professional athletes from other contact sports and 8 professional athletes from non-contact sports. This includes Gehrig and also Catfish Hunter. And interestingly, today is would have been Catfish Hunter's birthday. And many of you may know of Catfish Hunter because, again, his connection to the Bay Area is Oakland A for many years. It also has affected 7 politicians, including Jacob Javits and Mao Zedong, and 5 scientists, including Stephen Hawking. So is this list biased? It's a crowdsourced list, so it is going to be biased by society and the way we view things. So you can see that at least the people I've listed are almost all men. They are all men, rather. And if you look at the list carefully that's in Wikipedia, nearly all of them are men. So we know that there's a gender bias. There is a bias. Men are more likely to get ALS than women. They're about 1.5 times more likely, but this list is more skewed than that. But the other question is this link skewed? Is this list skewed because football players are more famous? Well, it's hard to know for sure, but there was a set of studies that I think might shed some light on this. So if there was a study done in the early 2000s, 2 studies done in the early 2000s. The first was done looking at soccer players. And this is a group of professional soccer players in Italy. There were 7,000 soccer players that were followed. And there was a 6-fold excess of deaths from ALS in these patients. So more than 1 per 1,000 players. For those playing greater than 5 years, the risk was 15-fold higher. And for those playing less than 3 years, the risk was only 3-fold higher. So it sort of suggests a dose response. In other words, the more you get of this particular risk factor, the higher your risk for getting the disease. And that is one of the things that we like to see when we're trying to associate causality with a particular risk, a particular factor. And the same group did a follow-up study on professional cyclists in Italy. So same group of people in terms of ethnicity and potentially in terms of genetics. And they found no patients with none of the cyclists had ALS. They were only able to follow about 2,000 cyclists. So maybe they didn't have a big enough group to capture, but based on the study of the soccer players, we would have expected them to find at least 2 cyclists if the risk was the same. So if this was physical activity driving this risk, we would have expected to see that. But maybe it's something different. If you think about what soccer players and football players and other contact sport players have in common, it may be that TBI, or traumatic brain injury, and it could be very mild repeated head injuries, may explain the increased rate of ALS associated with physical activity. So really what we're seeing is by screening for physical activity, we're actually capturing a lot of people who play contact sports. And also, as I mentioned earlier, military service is a risk for ALS. And that, if you think about what military recruits and people who are on active military duty go through, they're often more likely to have traumatic brain injury. And one way to think about this is maybe ALS is part of a spectrum that includes CTE, or chronic traumatic encephalopathy, which many of you have probably heard of. Interestingly, some of the pathological findings of ALS are seen in patients with CTE. So I'm going to move on now to talk a little bit about a machine learning project that we're involved in to understand the role of environment in ALS. And I'll walk you through this hopefully and explain it as we go along. So this is a collaborative project between a group that I'm part of here at Stanford in the VA. It includes Tom Osborne, Paula Suarez, Kevin Grimes, and Jennifer Wilson, and a Lawrence Livermore National Lab team headed by Amy Greischuk, Preet Ray, and includes Braden Soper, Jose Candenya, Sam Nguyen, and Jacob Pettit. We have funding. We're very grateful for the funding that we got from the Lawrence Livermore Lab Foundation. And that actually helped us to put together a really interesting proposal that is now funded by the Congressionally Designated Medical Research Programs, which is funding that's provided by the Department of Defense. So let me walk you through how we got into this project. So I'll give you a little bit of background on ALS and veterans and why we're involved here at the VA. So there's a twofold increased risk for ALS in veterans who were in the armed forces and deployed during the Gulf War. And in 2017, there's a large study that looked at people who had served in the military at any level in several different countries. And they found that there was about a 30% increased risk. So we estimate that the risk, if you're on active military duty, is between 50% higher and 100% higher than it is for the average person. So in 2008, the Veterans Health Administration, so that's the organization that the department that runs the VA, designated ALS as a service-connected illness. So what that means is they have determined that somebody who is in the military who developed ALS developed it in part because they served in the military. And because of that, the VA has provided financial and medical support to veterans with ALS. This has actually encouraged a lot of people who normally would not have gotten care through the VA healthcare system to start using the VA healthcare system because they get full support for the disease and also get some compensation from the Veterans Administration. The prevalence of veterans with ALS is estimated to be between 4,000 and 5,000. So at any point in the VA system, there are between 4,000 and 5,000 people who are being treated for ALS by our organization. The VA healthcare system is a really interesting system. It's the largest integrated healthcare system in the United States. We have an electronic health record system that dates back reliably to 1999. And in that healthcare system data set, we have details on diagnosis, treating specialty, procedure codes, prescriptions, lab results, vital signs, demographics, benefits, and mortality. And this VA electronic health records collection is really the largest and most comprehensive electronic health records data set for ALS patients in the world. So we thought it would be a good idea to take advantage of this. So we're leveraging machine learning to identify environmental risk factors and disease modifiers for ALS. And if you remember, I mentioned there's some caveats for people who are looking for environmental risk factors. The first is that the exposome is vast in time and space, and environmental research typically requires these time-consuming and invariably incomplete surveys. So what we wanted to do is use the electronic health record to identify environmental factors. So it's got to be something that's clearly identified in electronic health record. It's something that doesn't require patient reporting, and something ideally that hasn't been researched extensively in ALS. So we actually came up with an interesting topic to look at, and that is medications. So not medications that are used to treat ALS, but medications that are used for other things. So in other words, if you were given a particular medication, say for blood pressure, is that going to influence your risk for developing ALS later on? And you think, well, that doesn't make sense because that medication is specifically for blood pressure. But if you think about how medications are developed and how they're designed, they're really purified compounds that come from some other source. So anybody who's ever thought that diet might influence your risk for a particular disease must also acknowledge that medications, even for a separate disease, may influence the risk of getting a disease, maybe something like ALS. So what have we done? I'll give you a brief overview of what we've done so far. We've been able to identify and curate the medical records from 15,000 veterans diagnosed with ALS between 2010 and 2019. This is a huge number of patients, more than any other organization has for patients with ALS. We've actually also been able to perform an initial analysis to identify medications, demographic factors, and comorbidities that correlate with survival of patients who have been diagnosed with ALS. And we're now using causal modeling, and this is really driven by the group at Lawrence Livermore, to determine how these factors may be affecting survival. So in other words, let's say that we did find that a blood pressure medication influenced someone's risk for having ALS. Well, is that because the underlying problem, the blood pressure, increased their risk and they just happen to get a medication because of that? Or is it really the medication that's driving that risk? And that's where causal modeling comes into play, and where machine learning is really important to sort these things out. So that covers what I wanted to talk about in terms of risk factors for ALS. And I want to give you now an optimist view. I'm the optimist view of the future for ALS and for these other neurodegenerative diseases. So first, I wanted to reiterate that risk factors can be hard to find, but they are sort of low-hanging fruit. So let's say, for example, that one of these medications that we find actually influences or slows the progression of ALS. Well, that's a medication that's already been approved by the FDA and has been given to patients and apparently causes no other problems or a few other obvious problems. That's going to be something that we can start implementing in the clinic right away. And if you think about it, data is being collected on all aspects of our lives. As we become more digital in everything we do, there's more and more data being collected. Google knows more about me than I know about myself. At some point, someone may use that data to figure out why I get a disease 10 years from now. Fortunately, machine learning and supercomputing is going to allow us to efficiently sift through these huge data sets and allow us to identify environmental risk factors that we would have never been able to identify before. And these may be risk factors for the disease, but they also may be things that modify the course of the disease. So the last thing that I wanted to talk briefly about is this concept, or I've phrased it this way, nurturing nature. So I mentioned before that, there's certain things we're stuck with. We're stuck with our genes and we're stuck with aging, and we can't really do much about it, or at least seemingly can't do much about that. So I want to talk a little bit about insights from a mouse model of ALS. And this is work that was done in Aaron Gittler's lab at Stanford. He's a colleague and good friend of mine, and a graduate student in his lab, Lindsay Becker, had done this work. And what she did is she looked at this mouse model of ALS. And this mouse model is based on putting an extra gene. This is a gene that's been associated with ALS in people, and they put an extra copy of that gene into these mice. And normally a mouse would live over, close to 2 years without any problem. But these mice, they die within about a month. And they rapidly decline, and they look like they have ALS. If you look in their brains and their spinal cords, they have those TDP-43 accumulations that we talked about before. And they get that same weakness. They start to lose muscle mass and lose control of their legs. What Lindsay did, and this was based on some work that Aaron had done previously, is she took these mice and she crossed them with another mouse, and that mouse was lacking a different gene, a gene called ataxin-2 or ATXN2. And when she did that, so these are mice, they still have that gene that causes ALS, and they're missing a different gene. And now look at what their survival curve looks like. So this is how many are surviving. And you can see that, granted, many die after 30 or 40 days, but you've got some mice that are living almost 2 years to 2 years of age. This is incredible. And this is a quarter of these mice are living. And before, none of them were getting past 30 days. So a remarkable effect. You're like, okay, so that's a gene, that's a knockout mouse. You can't knock out genes in people. That's true, at least not yet. CRISPR may change that, but probably not in the way that's going to be as helpful as this knockout gene was in this mouse. But there is another approach to that, and that's something called RNA interference. So if you have DNA, and normally when we think about how DNA works, DNA is a blueprint for making proteins, and proteins are the things that really run things in our cells. So DNA undergoes a process called transcription and gets converted into something called RNA, and then that RNA is then translated to make the protein. There is these molecules that you can use called antisense oligonucleotides. So an antisense oligonucleotide is a piece of RNA that matches up perfectly, is complementary to this RNA. And because of that, it will form these bonds. And these bonds are very similar to what you see in the DNA, which is 2 strands of DNA. Here you have 2 strands of RNA that are complementary. But cells don't like to see double-stranded RNA. They get very upset with it. They think it's a virus, and they just chew it up. So with these antisense oligonucleotides, you can actually prevent the expression or the eventual translation of a gene into a protein. And that's what Lindsay actually did. So she took antisense oligonucleotides to ataxin-2 and injected it into the mice that had the gene for ALS. And you can see here that you again get this effect where several of your mice are living much, much longer. Okay, so again, this is mice, and she's injecting them into the mice, these antisense oligonucleotides in the mice. Is that really something we can do in humans? Actually, it is. There's actually a clinical trial going on right now for patients with amyotrophic lateral sclerosis with this antisense oligonucleotide to the ataxin-2 gene. There's several different locations for this studies being done, and I think that a lot of people are very excited about this. It may not cure the disease, but if it slows the progression of the disease down, this is a new tool, a new arrow that we'll have that we can start using against this disease and other diseases. So I want to summarize now. As I mentioned, Alzheimer's disease, Parkinson's disease, and ALS are unique age-related neurodegenerative diseases, but they share common traits of pathology. Age, genetics, and environmental factors influence our risk for these diseases. Moderate physical activity and maintaining overall health. So I mentioned before these cardiovascular risk factors and their effect on Alzheimer's disease and Parkinson's disease to some extent may reduce your risk for neurodegenerative diseases, and traumatic brain injury may increase the risk. And I think that the previous assumptions that physical activity are actually increasing the risk for ALS may be muddied by the fact that it may be TBI that's driving that. I think that machine learning is going to facilitate our discovery of new modifiable risk factors, whether you go about this through looking at electronic health records as we are, or actually looking at changes in gene expression in cell cultures of these cell culture models of these diseases, or even in mice. And I think that new technologies, things like CRISPR and antisense oligonucleotides, are going to help us ameliorate the risk associated with genetics that cause disease, and maybe even aging as we start to understand more about aging. So with that, I'd like to conclude, and again, thank the sponsors of this talk, and I'm now able to answer any questions you might have.
Thank you very much for that, a very informative talk, Dr. Reimer. One of the questions we got is a bit of a personal question. What made you decide to go into this field of medicine?
There are a couple of things. So I think that when I was in medical school, the brain was the most interesting thing to me. I was very interested in research, and I realized that at the time, so this was in the 1990s, neurology was way behind other areas like cardiology, and I thought there was a lot of room for improvement. So I felt that it was a fertile field to go into. And interestingly, as I've gotten older, I have family members that are affected by these diseases, and that's actually helped increase my interest in these diseases.
Okay. Well, and another question is, what are the most significant advances that have been made in the time that you've been practicing medicine? What are the things that you said you're an optimist.
Right.
What are some of the things that have made you an optimist?
So I think there are two things. So I think one is the revolution in molecular biology or genetics. So we now understand a lot more about these diseases than we did before in terms of what's the underlying genetic risk factor for these diseases. And that's, that gives us information, not necessarily actionable yet, but I think that it is these new approaches like antisense oligonucleotides and CRISPR that really is making a big change. And then the other thing is this recognition, I think, being very pragmatic about it. I'd have to say that the most important thing has been this discovery that cardiovascular risk factors are huge, important risk factors for Alzheimer's disease and aging, Alzheimer's disease and dementia. I think that's been really profound and has dramatically affected the way that I address and treat patients. So now the first thing I do is I start looking at people's cholesterol levels, what their blood pressure is, what their glucose is, before I do anything else if they're coming in to see me with dementia.
Okay. At one point you mentioned BP meds, and one of our listeners is asking, is there a type of classification of drugs you've already identified?
Nothing formally identified yet. We're in that process. We have some preliminary data that suggests that there are particular medications that seem to have an effect, but it's too early. Another important thing to keep in mind is that we have one population of patients that we're looking at, and really before, and this is in part because of all the caveats that I mentioned about these environmental risk factor studies, we really want to look at it in the second population before we even really say anything. Not because we don't want people to change what they're doing if we think it's going to make a difference, but we don't want to mislead people.
All right. Someone else in our audience asked, what about prions?
Yeah, that's a great question. So there are a couple of things about that. So prion disease, as some of you may know, I think the most famous is mad cow disease and Creutzfeldt-Jakob, but more people are familiar with the name mad cow disease. These are diseases that are really rare. The discovery of them is really important for a couple of reasons. So one is, it made us realize that proteins can be transmitted. So the idea is we think of infectious diseases usually require the transfer of DNA or RNA. So if you think about bacteria or viruses, they have nucleic acids in them, or these DNA or RNA molecules. Prions are the first diseases where all that was necessary was some protein. And so that was a really important discovery. But the other thing is it sort of suggested that there's a new model for thinking about neurodegenerative diseases, and that is the spread of the disease. So if you think about prion disease, you'll have one cell that gets infected with a prion, and that cell will then create a bunch of new prions that then can infect neighboring cells. We're now realizing that that same mechanism may be occurring in diseases like Parkinson's disease and ALS and Alzheimer's disease even. And I think I mentioned that patients with ALS, sometimes it'll just kind of start slowly marching up their spinal cord. And we think that's actually because of a prion-like mechanism of spread within their spinal cord. So if we can understand how prions spread, maybe that'll help us understand how ALS spreads, or how Parkinson's disease gets more severe, or how Alzheimer's disease starts to affect not just memory, but language and then vision.
Okay. Some years ago, creatine deficiency was a leading theory relating ALS to exercise. Yeah. Is that still considered a valid theory?
It's considered a theory. It has not been proven, and I think that most people don't recommend creatine supplementation. There's phosphocreatine and other things that you can take as supplements, and there have been trials to look at whether or not they influence the progression of the disease, and they don't, unfortunately.
Okay. And someone else wanted to know if you could expand on the role of hearing loss for Alzheimer's.
Yeah, it's fascinating. I think that and I usually try to come up with at least more than one explanation for something like that. And I have at least two. So one is that, it's the use it or lose it. So if you can't hear things, you're not communicating or interacting with people as much. And that kind of makes sense. And I mentioned that social isolation or increased social contacts decreases your risk for Alzheimer's disease, as decreased social contacts seems to increase your risk. So that may be what's going on. The other thing is, if you think about what hearing is, it's actually a neural process. So this involves, your inner ear receiving sounds and then converting them into a neurochemical process that then goes through your brain. So decreased hearing may actually be just a manifestation of a neurodegenerative disease.
A few years ago, there was a lot of publicity about diets that could even reverse the progress of Alzheimer's. What is the state of research on that?
So that's ongoing again, and I think this goes back to this issue of it's really difficult to do. It's difficult to do surveys to find out what people have eaten. It's even harder to tell people what to eat and make them eat that. So there's really no circumstance in where we can do the proper placebo-controlled study where you ask whether or not one diet influences your risk for disease. We do know that people whose overall health is better do better, both in terms of progression of Alzheimer's disease and in terms of risk of getting it. If you think about those cardiovascular risk factors that I mentioned before, we do know that they are definitely linked to diet. So high blood pressure, diabetes, and high cholesterol, all influenced by diet to some extent. And so diets that decrease your blood pressure, decrease your blood glucose, and decrease your cholesterol are likely going to be helpful in terms of reducing your risk for Alzheimer's disease.
Right. It seems there's been more attention on diet in relation to Alzheimer's than there has been on other diseases, ALS or Parkinson's. Have there been similar studies about diet and how it relates to the disease?
It's hard because of the smaller number of people that have this. So if you can get enough people in a study, if you can survey enough people, a lot of the noise will kind of be lost. You'll be able to drown out that noise with some signal. But when you only have 20,000 people with ALS in the United States, and not all of them are going to be able to be part of your study, or when you only have 500,000 people with Parkinson's disease, and certainly not all of those are going to be involved, you really can't get that data. I think this goes back to my point about big data and machine learning, really kind of maybe changing the lay of the land in terms of this type of research. So if you're tracking how someone's buying, what food someone's buying, you might be able to better determine what they actually are eating. So I'm on the low fat diet, but I went to McDonald's yesterday. It's one of those things where you could have more, better data.
Another of our member of our audience wanted to know what research has been done on smoking, caffeine, their addictive aspects to decrease the risk of Parkinson's disease.
Yeah. So again, this is like, I don't think any physician would ever tell you to start smoking for any reason, even if you had a strong family history of Parkinson's disease, because your risk for the complications of smoking, of getting heart disease or getting lung cancer, is just far too great. So these, again, are all arguably studies you can't do. There have been some attempts to look at caffeine consumption, but again, there are people that just don't tolerate caffeine, and are those people more prone to get Parkinson's disease later on? Maybe they're going to be the ones that drop out of the study that you're trying to do, and it becomes this really difficult thing. There are ideas like doing nicotine patches and whether or not those are going to influence your risk. Those are interesting ideas, and there are, I believe, some studies that are looking at that.
Another of our member of our audience said that his or her mother, Parkinson's symptoms get much worse with indigestion.
Oh, interesting. So it's interesting because indigestion is a common problem in people with Parkinson's disease, because one of the parts of the nervous system that's affected in Parkinson's disease is what we call the autonomic nervous system. This is a part of the nervous system that controls blood pressure, heart rate, but also controls how fast food moves through your intestines. So it may be that her indigestion is a sign that her Parkinson's disease is less well controlled. And the two go hand in hand, not as cause and effect, but as both being the same process.
Somebody else wanted to know your views on artificial sweeteners.
Yeah, good question. Not a lot of data. And it's really, it's a tough question, because I had mentioned that diabetes it increases your risk for Alzheimer's disease, and may increase your risk for Parkinson's disease as well. And, is aspartame or stevia or something like that, is that going to benefit you? And we don't know. And there are some recent interesting studies that say that have suggested that even artificial sweeteners have a weird kind of paradoxical effect on your body, and so they don't necessarily help you lower your blood sugar as much as you want, and they can do other things that are unanticipated. So right now, I'm sort of agnostic on whether or not one should switch over to artificial sweeteners. I would just argue for reducing simple carbohydrates.
Someone else wants to know if there has been any comparison or inclusion of data related to CTE.
So there are ongoing studies about CTE, and CTE is a clinical diagnosis linked to pathology. So it's a little bit hard to make, and it's really, if most of the studies are done at centers that specialize in CTE. So there's a large center in Boston, and they're doing a lot of the research on this. So they're defining the disease and making some important progress in that. And I did mention that the pathology that we see in CTE is very similar to the TDP-43 accumulations that we see. We see those in CTE just like we do in ALS. So I personally suspect that there is a strong connection between the two, and that what's happening in ALS, particularly if ALS is associated with traumatic brain injury, what you're seeing sort of a very limited CTE that's just affecting these motor neurons. And the spread then occurs, that prion-like spread can occur once it starts.
How about wine? Some neurologists call it a toxin, yet many of us live in wine country.
Yeah. So I think that someone said poison has to do with dose, and maybe that's the best way to think about it. So in moderation, there are many studies that suggest that healthy lifespan is extended by moderate alcohol consumption, but moderate is hard to define, and for each person is probably a little bit different. So just like no physician in the rightful mind would tell you to go out and start smoking, no physician in the rightful mind will tell you to go out and start drinking heavily. But I think in moderation, there's potentially some benefit, and I suspect, again, in moderation, no harm.
Mm-hmm. Another audience member would like you to expand on environmental factors, TCE and the Parkinson's epidemic.
Yeah, so there are the chemicals, including TCE, that have been that are thought to be linked to Parkinson's disease. And again, it gets very complicated because the data for this are not as strong as you'd like them to be. And certainly, again, you can never do a controlled trial to determine whether or not there's an actual effect. And TCE is everywhere. It's hard to find anybody who hasn't been exposed to it. And even if you were, say, to find accumulation of TCE at higher levels in people with Parkinson's disease, that doesn't mean that TCE is the only thing that's accumulating. It just happens to be the one that you see. So, I think that there's definitely benefit from limiting TCE exposure and reducing the amount in the environment. And it's for other reasons, because it's thought to be linked to other things as well. But, its role in Parkinson's disease, I think, is still, some people feel very strongly about it. I'm not as convinced as other people are.
Do you see clusters of ALS patients, perhaps with a link to a particular environment?
Yeah, that's a good question. So the closest thing is, arguably, is sports. So you do tend to see clusters in the sense that you've got a large number of NFL players that have ALS, larger than you'd expect. The problem with clusters is there tends to be a bias. So you tend to see clusters of things, even though they may be random. So you assume causation or some link, so there may be 3 people on a block that get ALS, and it just happens to be random. On some block, you're going to get 3 people that have ALS, but there are going to be thousands of blocks where no one has ALS. And it's not because that block happens to be in a particular location. But it is possible that there are some compounds that increase your risk for ALS that we are not aware of, something that may be in groundwater or something along those lines.
All right. Are proton pump inhibitors like Protonix associated with dementia?
Yeah, they are. As far as I know, there aren't any great studies that show that they are. They do affect absorption of certain things, and so they may actually have an effect, but there's no definitive data. If there was, there would be a giant New York Times article and a Washington Post article and San Francisco Chronicle article on how everyone should stop taking them. So unfortunately, there's some data that suggests they may, but not enough data for us to make clinical decisions about it.
Now, what other diseases cause muscle mass loss?
So there are a couple of different things. So the group of diseases that we call neuropathies. So neuropathy is damage to nerves, and it's pretty common in certain settings, particularly in people who have diabetes. Those nerves aren't attaching to the muscles, and when they don't attach to the muscles will start to get smaller. And you can also get local nerve injuries. So carpal tunnel syndrome, one of the most common things that we see in carpal tunnel syndrome is loss of muscle mass in the hand. So that's the neuropathies, whether they be compression neuropathies like carpal tunnel syndrome or ulnar neuropathy or something like diabetic neuropathy are probably the most common. We also see it in primary diseases of muscle. So myopathies, there are different forms of myopathies that can be caused by genes. There's some that are caused by inflammation, and there's some where we don't know the cause.
Is lack of sleep a risk factor for any of the diseases we've discussed tonight?
That's a good question. So there's ongoing research. There was a very interesting study that looked not at sleep per se, but at oxygenation during sleep. So this is a study that was done where patients underwent sleep studies to determine whether or not they might have sleep apnea. And those patients were followed up later on to see if any of them developed dementia. And there's a much higher risk for dementia in the group that had low oxygen levels during sleep, something that you would see in someone with sleep apnea. So we do know that sleep is important. Sleep apnea is a risk factor for developing dementia. And we suspect that disrupted sleep-wake cycles probably contribute to diseases as well, but not any great evidence. And again, this has to do with, you can ask me how much I slept last night, and I can give you a general idea, but I don't track my sleep. Well, now people can track their sleep. I actually don't do that myself, but you can get sleep trackers nowadays. And it may be that 5 years from now, we're gonna have some great data on how long you sleep and whether or not you get Parkinson's disease or Alzheimer's disease or ALS.
How might some of the things you discussed for ALS apply to Parkinson's disease, like RNA and the IA study?
Yeah, so the RNA, one of the things is to think about the genes that are associated with Parkinson's disease. And as I mentioned, GBA is one of the genes that's associated with Parkinson's disease. It's glucose cerebrosidase. That gene it's a deficiency in that gene that seems to be linked to Parkinson's disease. And there are actually now some trials where people are getting an extra copy of that GBA gene to see whether or not it affects their development of Parkinson's disease, or progression rather of their Parkinson's disease. So it's a little bit different than what I mentioned for ALS. For GBA in particular, and its relationship to Parkinson's disease, but there are other genes. So alpha-synuclein, that's the protein that accumulates in Parkinson's disease. The gene for that can be knocked down as well using RNAi, and that may be a great treatment for Parkinson's disease in the future. The thing about the difference between the reason this is progressing quicker in ALS is, ALS is a tragic disease with a really rapid course. Parkinson's disease, someone can develop Parkinson's disease and have for 20, 30 years. You think about someone like Michael J. Fox. And so it's a kind of morbid way to think about this, but ALS is actually an easier disease to test these new treatments on because the disease is progressing faster. We can see results more quickly. And to be honest, unfortunately, we don't have much to offer people with ALS. For Parkinson's disease, we do. We have medical treatments that help people function better. It doesn't change the course of the disease, but it certainly helps them function better. We have surgeries that do the same thing. So there's a lot to offer the patient with Parkinson's disease. For ALS, we really don't have that. So I think the community is being much more aggressive about developing novel treatments, and sort of proof of principle is going to come from ALS, but I think then apply to Parkinson's disease and later Alzheimer's disease.
Does exposure to aluminum or fluoride increase a person's risk of Alzheimer's?
Yeah, a great question. So there are a lot of studies that suggested there was accumulation of aluminum in the brains of people with Alzheimer's disease. The best study that addressed this was a study that looked at two communities in Kentucky. They had everyone in those communities was on well water. The concentration of aluminum was very different in the two communities. And one had low aluminum levels, the other had high aluminum levels. And the risk for developing Alzheimer's disease in those two communities was identical. So it really is, I think, good epidemiological evidence to suggest that consumption or contact with aluminum really has no impact on your risk for Alzheimer's disease. A similar study really hasn't been done for fluoride, but the risk for Alzheimer's disease seems to be universal and doesn't seem to affect countries or communities that use or don't use fluoride in their drinking water. So I suspect that it doesn't have a strong impact on that.
Is there a correlation between chronic inflammation and neurodegenerative disorders?
Yeah, that's a good question. That's an ongoing question. I think that there's a lot of evidence, at least from models of these diseases. So say, for example, mouse models, that inflammation and in the brain, so you think about inflammation in, say, the periphery, so in your arms, legs, or whatever, that's mediated by what we call white blood cells. In the brain, we don't have these white blood cells sitting in our brain substance. We have another set of cells called microglia, and these are sort of the immune cells of the brain. And they behave a lot like macrophages, which are the immune cells, some of the immune cells that we have in our periphery. And they really have a crucial role in progression of these diseases. So we're now realizing that they're really important targets for these diseases. So chronic inflammation is going to be an important player. How to control it, though, is another question. How important is it? Is it important to have some level of inflammation or be able to fight off an infection, say? We know that immunosuppressive drugs can cause real big problems in people, in the brains of people, so they can get certain types of infections or more prone to certain types of cancer. So we want to be careful when we think about it, but we definitely, I think everyone agrees that neuroinflammation and chronic neuroinflammation has an important role in these diseases.
Could you expand a little bit on the role of head injuries in terms of how it relates to the three neurodegenerative disorders?
Right. So these studies, these are epidemiological studies. So they're looking at the risk for looking at people with or without a particular disease, and then getting information from them about whether or not they had significant head injuries. And it's a really difficult type of study to do. But in each of these diseases, there are some studies that have shown that there's increased history or risk of concussions in people who have developed all three of these diseases. So we do think that there's some link between that. How that comes about, we're not really sure, but you had just mentioned this idea of inflammation. So what happens when you get a brain injury? Well, one of the things is you start activating those microglia, and they start, you have nerve cells that have been disrupted, and the microglia have to go in and clean things up. So they're definitely going to have a role in a response to traumatic brain injury. And that may be sort of the beginning of a process that leads to neurodegeneration later on.
And how do you recognize symptoms of Alzheimer's as opposed to differentiating it from other kinds of dementia without, when someone is still alive, presumably?
Right. So technically, the diagnosis of Alzheimer's disease is a pathological diagnosis. That means that someone has to look at the brain of the person and look for those amyloid plaques and tangles. We're developing better and better biomarkers for Alzheimer's disease. Most of them are CSF-based biomarkers or imaging biomarkers. So we rely on MRIs and PET scans, and then also analysis of spinal fluid that's collected. So that's one way that we can do it. The other is sort of the progression of symptoms. So all of these neurodegenerative diseases are really hallmarked by progression. So and the rate of progression. So if you walk into a room and you forgot why you walked in that room, that's pretty common. But if that's the first thing you notice, and then you start having trouble coming up with words, and then you start having trouble remembering how to get home from the grocery store, those are, landmarks of progression. And those are the types of things that we look for.
What is one thing you would like to know in order to better understand neurodegenerative diseases?
Ooh, that's a great question. What would I like to know? Well, I mean, to be honest, I'd like to know the answer to the study that we started, and that is, are there common medications that we're using that may influence the progression of these diseases? And there are two reasons for that. So the first is, immediate application of the results that we find. So again, if we found that some blood pressure medication influenced or slowed down the progression of ALS, that's great. We can use it right away. But the other thing is that knowing what that drug normally does may give us some insight into how ALS is progressing. So, and I'm doing this totally as an example. Let's say a calcium channel blocker, which is a blood pressure medication, slowed progression of ALS. Well, it would suggest that calcium channels may have a role in ALS, and that would be really important. So I mean, I study what I'm interested in, so I have to admit that would be where the question that I'd like to know the answer to. I mean, like everyone, I'd love to have a cure. I'd love to be able to close my eyes, open them, and have these diseases gone. And I wouldn't care how we got rid of them. They're horrible diseases, and they have tremendous effects on the people that are affected by it and their families. But the curiosity part, that I want to know part of my brain is like, oh, I'd really like to know if there's something here that we can use right now.
Is there a link between ALS and autism?
Not that's appeared in the literature. That's an interesting question. So if you think about autism, there's been this really big increase in the number of diagnoses of autism over the last several years. And we think that autism has something to do with the way that our brains set up and the way they wire. And we think the same is probably true to some extent for ALS, that miswiring, and true for any neurodegenerative disease, that aberrant wiring in the brain may be contributing to this. And it'll be interesting because this is, again, we're looking at electronic health records may have a huge impact on how we understand these diseases. So maybe now that there are more and more people who've been diagnosed with autism, that we'll start to see more people with ALS and be able to make that connection. But so far, no one's made a clear connection between the two.
Mm-hmm. Is stress
The question is stress a risk factor?
Oh.
Don has frozen up for
Yes.
a little bit, so
Yeah.
I'll step in.
Yep, that's a great question. So stress is hard to define, and again, I hate to keep on harping on this, but these studies that look for risk factors are often complicated because how do you define stress? So there are a couple of different things that you can think about. So we know that stress seems to be implicated in cardiovascular disease. So people who have stressful occupations or describe themselves as stressed people tend to be more likely to develop heart disease. So that may be a link. But beyond that, there hasn't been a lot of great evidence. I suspect that there is a connection to some extent. It's sort of like, the question about sleep. I really do think that if you're sleep deprived, you're more likely to get one of these diseases. And I really do believe that if you're under chronic psychological and or physiological stress, that this may have an impact on your ability to avoid these diseases. Let's say that the body has a mechanism or mechanisms to help us avoid these diseases. And those mechanisms are going to be stressed and not function as well.
Last question. Could you comment a little bit more on your collaboration with Lawrence Livermore? Sure. And especially with a view to perhaps there are other capabilities at the laboratory that would be useful to you in your research.
Absolutely. So the study came about in part because of Lawrence Livermore's interest in ALS and the fact that we have a large ALS clinic here. And so there are a lot. I think that the electronic health records is the beginning of what I hope ends up being a long-term collaboration with the Lawrence Livermore lab. There are a few things that I think are great to look at. So, where do we have large data sets that we can't sift through properly? So one of them is electronic health records. Another one is imaging. So if you think about it, a lot of people have gotten brain MRIs or spine MRIs for lots of different reasons. There may be hidden clues in those images that we can't pick up, that the human eye and human brain can't figure out. And I think that the Lawrence Livermore National Lab Group, they have the capability of not looking at this as an image, but as a collection of data, and be able to get through it in a way that no human being, not even a collection of human beings, a huge collection of human beings can do. So I think that's one thing that's really amazing. And there are other things that I'm not directly involved in that the lab can do that I think is really fantastic. So I had mentioned TDP43 earlier. This is that protein that accumulates in patients with ALS and in the cytoplasm. Well, why does that aggregate? I mean, the great thing about supercomputing and machine learning is that you can actually start modeling things and trying to understand how proteins that normally shouldn't stick together end up stuck together. And you can also start thinking about, well, what can I wedge in there to kind of separate these things out and start spreading them out? And you can model these things and make great advances. So this, I think Donna had mentioned this. I mean, there are great capabilities that the lab has that can be leveraged to start addressing these things.
Well, Dr. Reimer, thank you very much for a very informative and very clear-eyed yet hopeful presentation on what's a very difficult topic for a lot of people. We hope that perhaps you could come back in a few years and give us an update on where we are. So thanks again very much for presenting for us tonight, and we look forward to having you again sometime in the future.
Thank you very much.
So that brings us to the end of our formal presentation. I have a number of people I'd like to thank. We'd like to give a special thank you to Kathy Brown for her moving testimonial that helps us understand the full human dimension of this disease. Also, thanks to Donna Crawford and the Livermore Lab Foundation for partnering with us to present this live stream and provide an opportunity to support important scientific research. Only a team can make a production such as this a reality. So I would like to thank Karen Bennell and Uma Kale for their assistance tonight. I certainly couldn't do this alone. We'd like to thank the Bankhead Theater and Chris Carter for providing the platform for this evening's presentation. And we thank Eric Johnson, the wizard behind the curtain, for his management of all video aspects of tonight's presentation.
Thank you, Don.
You will. We hope that you will sign up to see the National Geographic presentation, Women and Migration, on April 14th, which we are co-sponsoring with the Bankhead Theater. We'd like to thank all of you for your continued support of the Ray Dorough Speaker Series. Good night, and please stay safe.